Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all, and welcome, and thank you, SynBYSS people, for joining us, the 103rd seminar, and I call it a UC Berkeley Day. I just got back from Germany, and I will go to Orlando this Saturday to attend the annual AICH meeting. I hope to see many of you there, although I would not have time to meet Mickey Mouse. Okay, it is my tremendous honor to introduce very briefly, but I'm not sure I can do so, our pioneer speaker, Professor John Dueber. He does not need any introduction because we all know him. He is a professor at UC Berkeley, the Agilent Biodesign Program co-chair, and also a trail runner. Importantly to me, he was my first collaborator who was not from the Kristalla Preder lab when I was a PhD student at MIT almost 2 decades ago. And he and I published a paper in Nature Biotechnology, the most cited paper among my papers, on protein scaffold for co-localization of metabolic pathway enzymes, and another paper in metabolic engineering, also on the application of protein scaffold. John, thanks so much for your pioneering work in metabolic engineering and also synthetic biology, and also great collaboration almost 15 years ago, and the virtual podium is all yours now, and thank you again.
Let's see. Is that
Yes.
shown correctly?
Yeah.
Great. Well, let's see. Well, thanks so much for that kind introduction, Tesa. It's too kind. I'm humbled to be called a pioneer in synthetic biology. I don't know if that's true. Might be more that I'm just old. And I can't believe it's been about 15 years since that collaboration. That was a lot of fun. Well, nonetheless, I'm honored to share the Zoom with our talented headline speaker, and I will try to keep to time, so definitely give me a warning if I'm starting to go over. With the briefness of this talk, I will just give a high level about one project that we've been working on in the lab for quite a while, and I'm really excited about, and that is repurposing the yeast peroxisome to be a synthetic organelle for the compartmentalization of engineered metabolism. And this is a project that has been performed and made possible by a lot of really talented graduate students and postdocs in the lab. So I'm going to start by showing their pictures and names. Obviously, I'm not going to talk about each of their individual projects that they worked on under this umbrella. But I wanted to start here because working with them has been the true excitement of being a faculty member. And they're all now, most of them have graduated on the slide, save a couple, and are off doing their own amazing things in the real world now. I also want to call attention to the funders of the project. Early on, Department of Energy Early Career Award that kicked off this project, and then NSF has funded a couple of projects, and I want to give particular attention to the NSF funded Center for Cellular Construction, which has been a great funding source that has allowed us to interact with fantastic labs and start thinking about this project from different perspectives that we wouldn't have thought about without this funding. So at the high level, why would we want to compartmentalize engineered pathways in a synthetic organelle? In our short-term goal, we want to be able to insulate our engineered metabolism from the rest of the cell or milieu. That could be just preventing crosstalk that would lose some flux of our pathway to undesired products, or in the extreme cases could cause toxicity that would be harmful to our production host. So in essence, we want to protect the cell from our engineered metabolism, as well as protect our engineered pathway from the cell. In a long-term goal, we'd like to be able to manipulate the chemical environment within this organelle so that we can make possible chemistry that wouldn't be viable in the cytosol. So why choose the peroxisome to make this synthetic organelle? It's obviously ambitious to try to make a synthetic organelle, so we wanted to start with the best platform possible. And after looking around, there's several choices that could be made, but we chose the peroxisome for a number of advantages that it relates. The chief one is that the peroxisome is not required by a yeast cell for viability. Here I've shown that we've, on the bottom cell spot, we've knocked out the ability of the peroxisome to import protein into its lumen. And you can see that the yeast are still viable. They're still able to grow. There's not a growth defect from this. Also, the peroxisome has been naturally specialized for different functions in various organisms. So in Saccharomyces cerevisiae, it's primarily used for the beta oxidation of very long-chain fatty acids, and that's why it's not necessary when it's grown on conventional fermentation media using glucose as a carbon source. But in these two industrial yeasts, they're used for protein production, Pichia pastoris and Hansenella pyomorpha, they compartmentalize the methanol utilization pathway. So if you grow these yeast on methanol as the main carbon source, the peroxisome morphology changes dramatically. They proliferate and become quite large to compartmentalize the methanol oxidation reactions that would be highly toxic if they happened in the cytosol. So we are interested in additional projects I won't talk about how this morphology change happens and can we use synthetic biology to control morphology changes. But I think for the purposes of this intro to the project, our foremost interest is that although it's ambitious to try to specialize the peroxisome for different engineered functions than what it naturally evolved for, nature has actually accomplished this already. So for time's sake, I'm not going to go into any of our individual projects, but I'm going to highlight two papers here if you're interested to look for more information. The first was one that we published in 2016 that lays out a lot of the tools that we engineered to start being able to repurpose the peroxisome. In particular, the ability to efficiently import protein and compartmentalize that protein. And associated with that, various assays for quantifying how well that compartmentalization is achieved. And then in the second one, in 2020, which we were lucky enough to have the cover art for, we used the peroxisome for the application of compartmentalizing a toxic enzyme so that the cell can be highly viable and grow well, but also maintain high titers with high expression of this toxic enzyme. And I will end there.
Wonderful. So that reminds me of your history or your career until now, because you start with yeast, engineer in Wendell Lim's lab, and at the time, I got to know you. And then you do E. coli work with me, and you're back to E. coli, yeast, but using synthetic biology tool for the metabolic engineering application. So that's absolutely amazing. And also the theme is the co-localization or protection of the enzyme, for the metabolic engineering application. So that's absolutely great. Thank you so much.
Thank you.
Yeah. All right, so now the main speaker of today with a longer introduction. Dr. Alissa Bleem holds a bachelor's degree in chemical engineering from Montana State University and a PhD in bioengineering from the University of Washington. She currently leads synthetic biology and lignin valorization project as a postdoctoral researcher at the National Renewable Energy Laboratory under Greg Beckham's guidance. As you know, Greg Beckham is one of the leaders in lignin valorization, and she is passionate about finding biological solutions to energy problems. And I believe she will talk about her recent publications on lignin valorization among many projects. I have been working on lignin valorization for more than a decade, and I am so excited to listen to her presentation today, although I listened to her talk in person at some conferences recently. Alissa, thanks so much for your time today, and please take it away. All yours now.
Great, thank you, Tae-suk, and thanks, John, for setting the stage. It's an honor to have a chance to present for you all. So hopefully my screen will share. Everything look good? Great. So yeah, thanks again for the introduction, Tae-suk. I think I sent you kind of a slightly outdated, or I sent you the kind of info about myself a while ago, because I'm not a postdoc anymore. I'm actually a staff scientist here at the National Renewable Energy Lab, but still working with Greg Beckham. So really haven't changed that much in terms of my role, just a different title. So today I'm going to be talking about some work that we've been doing to optimize aromatic catabolism for bioconversion of vanillate using evolution. So before I start, I want to thank all the folks and funders who have been involved in this project. This project was fully funded by the Center for Bioenergy Innovation, which is a BER center as part of the DOE. It's a really well-run center based out of Oak Ridge National Lab. So we have several collaborators there, including Adam Guss's group and Bob Heddick's group. And then we also have collaborators helping us do evolution at UCSD and DTU from Adam Feist's group. We're doing a bit of machine learning with Bernard Paulson and Hung Yu Lim to learn from these evolution data sets. And then in my group here at NREL, Greg, of course, is the leader of our group, but I've gotten a lot of help on biochemistry from Eugene Kowatscha, another staff scientist, and then technicians and interns, Morgan, Zoe, and Ian, who did a lot of experiments for this. Okay, so let's get into the talk. So what do we do here at NREL in our group? Our goal is to develop sustainable materials and products by combining biology, chemistry, engineering, and material science. So as Tae-suk mentioned, Greg has quite a large group, quite a large sphere of influence in the renewable energy space, but I'm going to kind of try and hone in on what my little sector does. So basically what we're trying to do is leverage engineered bacteria to take waste feedstocks, so things like biomass or plastics, deconstruct those and depolymerize those using catalytic, both chemocatalytic and biocatalytic approaches. And then what we get is like this mix of different, usually monomeric compounds, so things like sugars, organic acids, and aromatic compounds. And then where I come in is trying to engineer organisms. Oftentimes this bacterium, Pseudomonas putida KT2440, to be able to biologically funnel these mixed substrates that we get from waste feedstocks. So this term biological funneling basically means engineering an organism to utilize all of the aspects in a stream, which may be quite chemically diverse, and then enzymatically funnel those to a set of one or more key intermediates that can then be upgraded to value-added compounds. So these intermediates tend to be things like protocatechuic acid or pyruvate or catechol. And And then the value-added compounds are things that we can make into value-added products. So beta-ketoadipic acid or muconic acid are things I'm showing here. We then can hand off our fermentation broths to bioprocessing teams to do separations and upgrading with more chemocatalytic approaches to make things that are drop-in commodity chemicals or performance-advantaged products. So it's really exciting to be part of this whole workflow in Greg's group, and I really enjoy kind of being here in the middle of the process. So today I'm going to talk about a specific aspect of this biological funneling that I've been working on. Mostly I did this work in my postdoc. So when we're talking about utilizing biomass, lignin is a really big fraction of biomass. So the sugars are pretty easy to pull out using enzymatic hydrolysis, but lignin is this very recalcitrant polymer, right? It's super heterogeneous, very many different substituents in lignin in terms of the chemistry, but there are approaches to be able to depolymerize it. And when you do that, what you get is this slate of very heterogeneous aromatic compounds. And a lot of these aromatics have O-methoxy groups, like these shown here. So typically in lignin, you'll get different groups that have 1, 2, or 3 substituents, and the methoxy group can be on 1 or 2 of these. And in order to be able to biologically funnel these compounds in Pseudomonas putida, what we have to do is O-demethylate these groups to intermediates like protocatechate or gallate type acids, because only these compounds can be ring cleaved and then funneled to centrometabolism or to other products. So this O-demethylation step is really critical for being able to valorize lignin biologically. There are 3 known enzymatic mechanisms that can conduct this demethylation reaction in vivo, and those are shown here. So on the left, we have the risky non-heme monooxygenases. These enzymes work by activating molecular oxygen with a free iron and using a flavin group to shuttle electrons, ultimately releasing formaldehyde as a byproduct and the demethylated substrate. So an example of this is the Vanillate enzyme from Pseudomonas putida. That's a native enzyme. And this is just a model of that structure. In the middle here is a second mechanism. These are tetrahydrofolate dependent enzymes. These work quite differently. So in this case, the organism has evolved a mechanism where tetrahydrofolate can actually abstract the methyl group onto a specific residue, and then this can be recycled through C1 metabolism. So in this case, we don't get any formaldehyde released as a byproduct. An example of this enzyme is the Lig-M demethylase from Sphingobium SYK6, which is a well-characterized lignin-degrading bacteria. On the right, we have the third mechanism. So these are cytochromes P450, and these are really similar to the iron monooxygenases in that they use oxygen, but instead the oxygen is activated here by a heme group. So an example of this that our group has worked with extensively is the GCOAB enzyme, which is originally found in Mycolotopsis, but there's also homologs in Rhodococcus, as Taysok will know. So today I'm just going to talk about these 2 mechanisms because they're quite different from one another, and both of these mechanisms have activity on vanillate, which is the substrate that we're going to be talking about today. So we know that vanillate demethylation is actually a bottleneck in native Pseudomonas putida catabolism. So on the left here, I'm showing a plot of substrates and products during fermentation of ferrolate with an engineered strain of Pseudomonas putida designed to accumulate mucinate, this value-added product. And you can see that over time, the strain is accumulating mucinate by consuming ferrolate, but vanillate also accumulates if we utilize just the native Vanillate cassette. And this causes problems in terms of yield and titer and everything else. We know that if we simply overexpress a second copy of the Vanillate gene, we can actually overcome this bottleneck. But there's a lot we don't understand about the fundamentals of vanillate catabolism in Pseudomonas putida. And if we want to really push titers, rates, and yields in these systems, we'd like to be able to feed a lot more ferrolate or vanillate to the system. And to do that, we need to have really robust demethylation mechanisms. So that's what this project is really about, understanding native and engineered Pseudomonas putida catabolism of vanillate to make it better for engineering applications. So to optimize vanillate catabolism in putida, we wanted to look at these two enzymatic mechanisms that I talked about. So VAN-AB is the native mechanism, which the strain has evolved and presumably is pretty well regulated within the strain, but it does produce formaldehyde as a byproduct, which is toxic to cells. So we thought it would be nice not only to look at evolving strains that carry VAN-AB, but also those that are engineered to use Lig-M instead, because this system utilizes tetrahydrofolate and doesn't make any formaldehyde as a byproduct. So I don't have time to get into it today, but I was able to make a few strains for this project, one of which utilizes this Lig-M enzyme. And this required overexpression also of some native C1 enzymes and modifying the regulation in putida of C1 metabolism. But the strain does grow. And we compared that to a wild type strain, so just the native Pseudomonas putida KT2440, and then also a strain of KT2440 that had the native VAN-AB locus knocked out and then overexpressed under the strong PTAC promoter at another location. So I'm going to refer to these three strains as wild type or WT, VAN-AB plus, and then Lig-M. So you can see here, this is the growth profile of these three strains on 20 millimolar vanillate. Overexpressing VAN-AB gets a little bit higher growth rate than the wild type, as you might expect. And this Lig-M strain really struggles to grow, but it does eventually get there. So from here, we wanted to use adaptive laboratory evolution, or ALE, to see if we could improve the vanillate catabolism in these strains. So this is with the help of Adam Feist's group. Josephine Johnson and El Said Mohammed conducted these evolutions using a robotic system at the DTU facility. And what they did is for the wild type strains, since these grow pretty well, you could see on the previous slide, for the wild type and the VAN-AB plus strain, we just did ALE at a fixed concentration of vanillate. So 20 millimolar vanillate. And basically what you're doing in ALE is you're passaging the strain over and over. So thousands of generations of cells are growing in this condition. And over time, the idea is that the strain will naturally adapt to the conditions just through random mutation. So basically you passage from one flask or tube to another at a fixed concentration over the course of 60 days, and you periodically take samples for next generation sequencing. For the Lig-M strain, since this one grew really slowly, we actually started at a lower concentration of vanillate and then slowly increased that over the course of evolution to push the strain further. So we started at 5 millimolar and eventually got up to the 20 millimolar that was used in the top two. And as I said, periodically we're taking samples from this for whole genome sequencing so that we can see what the genomic changes are that are causing improved growth phenotypes in the population. So here's just a profile of the different trajectories of this ALE. So ALE works. It allows your strains to grow faster. So in the top left, you can see these are each of the 6 lineages that we evolved for the wild type strain. Started at a growth rate of around 0.3 per hour and got up to about double that by the end of the evolution. Similar was true for the VAN-AB plus strain, although these lineages were a little bit more variable. And then the Lig-M strain has a little bit different plot here. So this is just one lineage where the vanillate concentration is shown in orange, and then the growth rates are shown in blue. You can see that we started to bump up that vanillate concentration once we saw jumps in the growth rate. And then the arrows here are just locations where we took samples for genomic sequencing. All right, so these are the populations after the end of evolution. And again, ALE works. So we saw improved growth for all of our lineages and all of the strains. So in each of these plots, the parent strain is the black line, and then each of the colored lines is one of the population lineages. So you can see that in a lot of cases, we got really big improvements in the lag time for the strains. So essentially no lag in some of these. They just start growing right away in vanillate. And in some, we also saw improvements in the growth rate. For the Lig-M strain, this was really, really drastic. So for the two that had Van-AB as the demethylase, the improvements weren't quite as striking, but they were there. For Ligem, it was really massive. So giant improvement in lag time and also improvements in growth rate. This is just a plot of those improvements. So again, you can see that most substantially, we are improving the lag here as opposed to the growth rate. And that has a lot to do with the way the ale is set up. So essentially what we're doing is we're letting the strains grow in a tube until they hit a certain OD, and then we passage them. So that you can imagine that that setup kind of selects for strains that hit a certain OD first, i.e. have a shorter lag. So that's mainly what we obtained from this evolution, particularly in the 2 strains that had the risky type mechanism. The Ligem strain, as I said, evolved the most impressively because it was struggling the most at the beginning. And actually, we start to get growth rates and lag times similar to wild type, which was really exciting for a strain that used a completely different mechanism enzymatically to get through that bottleneck. So you can see in orange here, again, this is the parent Ligem strain, takes 120 hours to start growing. In black is wild type Cotida. In yellow are 2 of the best evolved lineages. So the lag time here you can see is about the same as wild type. And the growth rate isn't quite as good as wild type, but actually the growth rate is quite similar to Sphingobia SYK6 under the same conditions. And if you recall, this THF Ligem enzyme comes from Sphingobia. So it's interesting that we're able to achieve a similar growth rate by engineering in that mechanism in Pseudomonas putida. All right, so we know that the strains grow faster when we evolve them, but why? That's where the genomic sequencing comes in. So on the left here, I'm showing just a table of the different types of mutations that we saw in all of the lineages. So you can see for wild type and Van-AB, we had pretty similar number of mutations. So unique means that it occurred in just one lineage and then observed as the total. For the Ligem strain, as you might expect for something that improved so drastically in terms of growth, we saw more mutations. On the right here, I have a plot, a Sankey diagram showing the different converged mutations. So things that we saw consistently across lineages. And you can see that while there is some overlap in the types of mutations observed in Van-AB plus and wild type, the Ligem mutations were actually entirely distinct. And I think that's really interesting because these 2 have different O-demethylation mechanisms, and they appear to have evolved in a different manner. So the red stars here are showing different mutations that were also observed in our midpoint sequencing. So some things mutated early on in evolution and persisted through the entire evolution, whereas others were actually lost by the end. And then these other ones maybe took longer to appear. So we'll get into more details about these mutations now. Overall, though, these evolved strains have not only improved phenotypes in terms of growth on Vanillate, but they also have these kind of non-intuitive mutations that improve performance. So in some cases, we'll talk about this a bit later, the mutations are somewhat intuitive, just getting more of an enzyme or something. But in other cases, we have no idea what the mutation is doing, but it does seem to improve growth. So this gives us a launch point for more investigations, right? Looking at omics studies to see how protein or transcript abundance changes in the evolved strains. And then also, it gives us a point to reverse engineer from. So we can look at individual mutations in isolation and clean backgrounds and figure out why the strain mutated in such a way. So that's what I'll get into now. So the first mutation I want to talk about was observed very consistently across Van-AB plus and wild type strains. So the strains that use the Van-AB mechanism. And these mutations were observed in this gene called FGHA, which is an S-formylglutathione hydrolase. And this enzyme is involved in the glutathione formaldehyde detoxification pathway. So if you recall from before, this Van-AB type enzyme produces formaldehyde as a byproduct. And it was nice to see that we were getting mutations in formaldehyde detoxification in these strains. So FGHA actually acts kind of at the end of this pathway, but the detoxification works basically by glutathione binding to formaldehyde and then a formaldehyde dehydrogenase, oxidizing that product to make this S-formyl glutathione, which FGHA then hydrolyzes to formate and regenerated glutathione. So what are the mutations that we actually saw? They're shown here. In wild type, we saw mutations to this lysine and glycine residue. And then in the Vanillate plus strain, we saw these three mutations. There's not a structure of FGHA from KT2440, but there is one from a related cold-loving bacterium. And so if we make a model of the Pseudomonas putida sequence on that structure, which has 67% identity, we can get a sense of where these mutations lie. And we know that this enzyme needs to form a dimer in order to be active. And actually, all of the mutations we observed are predicted to lie along this dimer interface, which I think is quite interesting. This G258D mutation can form a salt bridge when mutated to the glutamic acid residue with the lysine over here. And then down here, this tryptophan to cysteine mutant can actually form a disulfide bond with the neighboring cysteine on this beta strand. So, of course, we don't have the structural biology from Putida directly, but even these predictions were pretty exciting to us, and we wanted to dig into them in reverse engineered strains. So to do that, first, we just put the mutations back into clean backgrounds. So these are the parent strains not evolved with single point mutations in FGHA. And the results are pretty striking for growth with exogenous formaldehyde. So you can see on the left here, if you grow the strains in glucose alone, they all grow the same. But if you grow them with increasing concentrations of formaldehyde, you get a protective effect from these single point mutations. So the dashed lines here are the Vanillate plus and wild type parent without any formaldehyde. And then the solid lines in red and black are with formaldehyde. And the other colors are the single point mutations. And you can see that all of these help the strains persist a little bit better than their parent counterparts. I think this is really striking for just a single point mutation in the protein. Similar is true as you go to higher formaldehyde concentrations. Actually, the Vanillate plus strain doesn't grow at all, but some of its mutants do. We can also measure formaldehyde in these conditions. So we'll grow the strains in glucose with or without formaldehyde. And after 36 hours, we can measure the formaldehyde concentration using a colorimetric assay. On the left, as you would expect with glucose, none of these strains have much formaldehyde present, basically none. But on the right, when we have exogenous formaldehyde, both wild type and Vanillate are not able to tolerate it very well. There's still a lot of formaldehyde here. But then our two best mutants, G258D and W15C, are essentially detoxifying the formaldehyde to levels comparable to those where no formaldehyde was added. So that was really a nice result here. We wanted to know what exactly was causing the increased performance. We can make some hypotheses based on the structure, but we wanted to actually do biochemistry to measure that. So Eugene Kowalska set up a nice assay where he could attempt to assess this. And unfortunately, S-formyl glutathione is really unstable. You can't buy it, and even making it with a formaldehyde dehydrogenase is pretty transient. So instead, we used a surrogate that's pretty well established in the literature. So 4-nitrophenyl acetate was used as the surrogate, and when you apply FGHA as a hydrolase in this system, you get a colored product, and you can use that as a readout. So here are the kinetics from Eugene on the right. You can see that the catalytic efficiency of the two best mutants in vivo, W15C and G258D, is about 2-fold higher than the wild type. So this is a really nice explanation mechanistically for why these mutants are better. And I think it's so cool that the strains can figure out how to make these mutations kind of on their own and select for them in ale. Okay, so now I want to move on to another example of mutations that we saw both in the Vanillate plus strain and the wild type strain. And this was mutations in a LYSAR type transcriptional regulator with the gene ID PP3493. So what is this regulator? Well, we know it's a LYSAR type regulator, and these regulators tend to regulate the gene that's divergently transcribed from them, so pointing the other direction. Unfortunately, PP3494 is a PUF. It's a protein of unknown function. So we really don't know what this gene is doing. But what we do know is that knocking out both the regulator and the PUF is bad for growth on vanillate. So here I have plots for growth of these knockouts on glucose. Everybody's happy. But on increasing concentrations of vanillate, knocking out the PUF in blue or the regulator in maroon is bad. And since knocking out the regulator is actually more detrimental than just knocking out the PUF, that indicates that this regulator has influence on proteins besides just its adjacent one. So we wanted to dig into that a little bit more. I'll also just add that in some work Andrew Borchert and I did on stress tolerance in Pseudomonas putida, we saw that this PP3493 gene had decreased fitness during growth on vanillate. This was a T-and-seq based study. So it's kind of nice to have some evidence corroborating the importance of this gene. All right, so digging in a little bit more into the actual mutations we observed in ale. So there were 4 mutations that we saw, 3 different ones in the wild type lineages and 2 in the VAN-AB+ lineages. Interestingly, this proline to leucine mutation was observed in both. And I think that's so nice when you see a convergence across different lineages, but also different strain backgrounds on kind of a single mutation that's most beneficial. So we analyzed that one in more detail, again, with just growth. So here, again, glucose, everybody grows the same. But on vanillate, if you have this single point mutation, again, clean background, just the proline leucine mutation, you get a lot shorter lag time and also a little bit of increase in your growth rate. And I've just shown, again, the knockout here for comparison. So we're still not quite sure exactly what this mutation does. We do know it's beneficial, but we're running transcriptomics now in the single point mutation strains relative to the wild type, as well as the knockout, to figure out what this regulator is doing and why it's so beneficial for growth on vanillate. Okay, a couple more mutations I want to dig into. This one I thought was really interesting because it was right in the mechanism that we were trying to probe. So we observed only in the midpoint samples, not at the endpoint, but just the midpoint. For several lineages, we were seeing mutations in the VAN-B reductase. So this is the protein in this risky enzyme complex that actually shuttles electrons to the monooxygenase to conduct the demethylation reaction. And what we saw was a couple of different point mutations in VAN-B itself. And this is so nice when this happens in ale. You know, you're trying to probe this specific demethylation reaction, and then the strain mutates right where you want it to. So this is awesome for us to be able to study mechanistically. Once again, we started by just determining the effect of these mutations in clean backgrounds in terms of growth. So on glucose, all the mutants in wild type grow the same. With vanillate, though, we see that one of these mutations, A24P, has a lot better growth than the wild type and the other mutation. We're actually not sure why this other mutation showed up. It doesn't seem to be beneficial. But definitely the alanine to proline mutation is beneficial here. So again, we did a little bit more digging on this. We can see that when we grow these cultures in flasks, vanillate is consumed much more quickly by the mutants than by the wild type. And again, Eugene did some nice kinetic studies to try and figure out if VAN-B was just a better catalyst. So this is assaying NADH utilization and NADPH utilization with VAN-A for both the wild type and the mutant. And interestingly, for both cofactors, the A24P mutant is actually a much less catalytically efficient enzyme. It also has a lower TM, so it's not more stable. But the strain's clearly selected for this, and it clearly is beneficial to growth. So the last thing we wanted to see was, well, is there just more of this protein around? Does it just translate better or have more transcript? And it looks like that's the answer. So even though A24P is a worst catalyst, it actually has a higher transcript abundance than the wild type. So this is kind of a weird plot, but basically it's measuring both proteins overexpressed relative to the native level of expression. So if you have wild type Van B, you have this much transcript, but then if you have the mutant transcript, you actually have even more. So it appears that the strain selected for this mutation on the basis of protein abundance or translatability as opposed to kinetics. All right. Lastly, I do want to get into a little bit about the mutations of these ligam lineages because there were so many of them. And most of the things that we saw in the ligam strains were actually not very intuitive at all. So we saw a few things like mutations in the flagella, deletions there. That's really common in ale. If you're mixing your cultures well, you don't need a flagella anymore. So it often kind of gets deleted in some manner. But a lot of these we didn't really understand. One that was quite nice, again, to see was that the demethylase region itself had mutations. So here we were seeing really massive mutations, sometimes 12-fold or 16-fold duplications of the ligam region. So we thought, okay, well, these strains are growing better simply because they have more copies of ligam. So let's test that in vivo. Here you can see in black, the wild type KT2440, in green, one of the evolved ligam lineages, in orange, the ligam parent, and then in blue, the ligam parent strain with a plasmid that overexpresses ligam, so extra copies of ligam. And this strain grows a little bit better, but definitely not as well as the evolved strain. So what we can see is that although having more ligam enzyme around does seem to be important, all of these other non-intuitive mutations in the ligam lineages are actually more responsible for the improved growth phenotype than just ligam amplification alone. So that has motivated, again, transcriptomic studies. We're basically comparing the transcriptome of all of the evolved lineages to the parent to try and figure out what sort of changes are occurring in these evolved isolates that are making them grow better. All right, so wrapping up, obviously, adaptive laboratory evolution can give you strains with improved phenotypes. So if you select long enough in a condition, the strains will naturally mutate to have improved catabolism. And in this case, it was for Vanillate. I think this is also just a nice mechanistic study for comparing Vanillate odemethylation pathways in Pseudomonas putida. It's pretty clear that Van-AB is preferred in these strains, but it's nice to know that we can engineer other pathways like the ligam enzyme and then evolve for improved performance that way. It also gives us non-intuitive targets that we can reverse engineer in clean backgrounds. So everything that I showed today was super mechanistic and just investigating the mutations and their performance in the wild type strains, but we can put these into production strains, right? So strains that are taking these waste feedstocks and converting lignin into products might benefit from these mutations that we observed in ale. And overall, I think this really expands our knowledge of fundamental metabolism in KT2440, particularly for these genes that had not really been annotated before. Going forward, like I said, we're doing some transcriptomic studies to illuminate the changes in physiology between the evolved and parent strains. And yes, we're going to put these into production strains as well. All right, so with that, I hope everyone learned a little bit more about how evolution can teach us about metabolism. I certainly did through the course of this project. And I'd be happy to take any questions. Before I do, I just want to mention that our group is nearly always hiring motivated postdocs. So if you have interest in this type of work, synthetic biology, separations, bioprocessing, feel free to give me a shout. I'd be happy to answer questions about that. Thank you.
Fantastic. So I want to say one thing first. Join, NREL, why not? Actually, I sent one student after his graduation to become that. So fantastic. So before, I asked some question I have. John, if you are still around, do you have any comment or questions?
Oh, great talk.
Yeah.
I guess I had one just general question about the ale. Do you have any measurement or idea of what the mutation rate and the diversity that you're able to sample is? Like how expansive is the search of mutations that then you were able to isolate the mutants that you showed? Is that representative of a really broad search of all amino acid changes that were possible at each position, or is it a pretty sparse search?
Yeah, that's a really good question. I'm afraid I don't have a great answer, but I think you've inspired me to try and look and add that to this paper. This is pretty much all unpublished, but hopefully it'll be a Christmas present to us to get it out. But yeah, I'd love to be able to include a mutation rate, natural mutation rate for Putida. I personally have not calculated that or explored that, but I think it's something we could find out. Certainly, we and Adam Feist's group have done a lot of evolution in this organism, so it's something we could probably try and calculate based on passages and mutations observed. That said, we're not really doing anything to induce or encourage mutation. We're simply selecting. So I really don't think that we're exploring the full sequence space here. We're just kind of exploring the sequence space that's accessed by natural mutation.
Right.
And one really exciting thing that we're going to start doing, hopefully with a new postdoc in the new year, is pulling together all of these ale data sets from Adam's group and maybe from others who have done this type of work to examine whether Pseudomonas putida has regions of its genome that are particularly mutation prone. So we have a student coming in who's an expert in machine learning, and he's really excited to analyze these data in many aspects, but that one I think will be an exciting one to just find out if there's kind of natural spots, hotspots of variation. That doesn't really answer your question, but
no, that's great. Actually, I have one more question, if it's okay, Tae-Suk.
Yeah, please.
So I was thinking during your talk, probably one really nice feature of the ales is that you're selecting in the condition you're actually interested in. So the mutations that you get probably don't have some deleterious side effect like a pleiotropy, and you're losing something that's desired because you are doing the selection in what you're ultimately interested in. So I wonder, do you think that makes it more likely that you can combine these mutations together and still not get a loss of that desired function, and then do ales again, and even if there is some loss of, some hit, you could get compensatory mutations somewhere else?
Yeah, great questions. I didn't show it very well in this presentation because I was trying to kind of depict the mutations that were seen most commonly. But each lineage and isolate has a set of mutations, right? Some more than others. And there were things that we saw very commonly paired in a single isolate, and then things that weren't. So we didn't do a whole lot of stacking mutations. We did for some of the point mutations. So in that formaldehyde detoxification enzyme, we put all 5 mutations into a single protein, and that strain grows horribly and it has very slow kinetics. So in that case, combining everything wasn't a good thing.
But if you did ales on that strain, I wonder if you would pick up compensatory mutations elsewhere that would rescue that growth.
Yeah, and I think that'd be really nice to kind of dig back through the data for, because we do have these midpoint sequencing times and the endpoints. So there are things that disappear by the end, or become more common by the end, and those might be exactly kind of what you're getting at there, things that compensate. So yeah, great thoughts.
Yeah, very cool stuff.
Thanks.
John, do you have any other question?
I think I'll hold. I think that I see there's a couple questions.
Yeah.
Okay,
so I'll
hold. I don't want to dominate the Q&A.
Sure. I also wait until this one answered. Interesting question I got from Allison. Were there any geoterm or EC number groupings in the unique LIGM mutations that indicate what the non-intuitive mutations might be doing?
Yes, okay, I'm reading that now. So yeah, that's a really good question. It's not super conserved. I think it definitely merits going through and doing more of this type of annotation analysis. That's a really good idea, Allison. Thank you. That's something we're definitely going to do with the transcriptomics data. So we have data sets for each evolved population relative to the parent. And in that case, definitely we'll be looking at Go terms and enzyme types that are elevated in terms of transcript level. But for the mutations themselves, that could also be worth doing. So thanks. We haven't really analyzed that, but we certainly can.
Okay, so another question from Yuju. Following John's question, is there any method to accelerate the ALE process?
Great question. So I highly encourage you to look at Adam Feist's papers on this. They're really experts in automating ALE and kind of thinking about how to set it up. So I'll say two things on that point. One is you want to be really mindful of the way that you set up the selection. So as we were just talking about, the mutations that you observe in these experiments are really a product of the condition that you're growing in. So we wanted to select mutations that improved catabolism, not, for example, tolerance to vanillate. So we had to pick a concentration of substrate that was able to be utilized, but not stressful to the strains. So doing things like that is very important for getting quote unquote good results, or results that respond to the conditions that you want. Another thing that you can do to actually accelerate the process is something that Adam and some of his collaborators recently wrote a perspective article on. And I think they called the title of it, "Have you tried turning it on and off again?" And basically, this is thinking about other ways that you can set up ALEs to explore a greater region of the mutation space. So if you're using just a fixed concentration of vanillate for the entire thing, you can imagine kind of like an energy landscape where you might be sitting in a well that entire time. And maybe you get over one hump into another well where you have like improved growth or improved mutations, but it might be hard to get into the biggest energy minimum, if you will, or the deepest well. And so to do that, Adam and others have proposed ALE techniques where you actually, for example, if you're going for tolerance to a toxic substrate, you shift the ALE between a concentration of high toxin to low toxin and back and forth. And by doing that, you give the strains a chance to kind of jump into new areas of potential mutation space that they wouldn't see if they're just at a fixed concentration of the toxin. So approaches like that are being proposed, but not super well validated, what I've seen so far from the literature. So I think, yeah, it's something that Adam and his group are certainly exploring now, but I think the theory of it sounds pretty good, but we'll see if it actually speeds up ALE in practice. Thanks for the question.
Fantastic.
Sorry, the theory of that is just to have more replication cycles at the lower inhibitor concentration, so that more replications per unit at a time, so that accelerates the mutation rate.
And then you might get some mutations that aren't necessarily relevant to that substrate that potentially open new spaces for compensatory or maybe not even beneficial, but different genomic backgrounds, right? Mm-hmm. So we'll see if it works for them.
Fantastic. So I asked one very challenging question to you. The evolution, I believe many people in this audience and us interest in, evolution, I guess. I recently, wrote an opinion article. The title is "7 Governing Principle in Biology." And obviously I chose one of the principles in biology is evolution. And obviously I believe everyone agree with me. But evolution is interesting thing. This is the double, side of a knife or whatever. Sometimes very beneficial. As you know, I rely on evolution, adaptive evolution for almost decade to modify the strain. You did same thing, but at the same time, this is the one of the challenging aspect of the biology or any biology biological engineer mutation is some issue always we need to deal with. So having said that, you used adaptive evolution to improve strains, but at the same time, I work on metabolic engineering almost 20 years. I wanted to see more engineering, forward engineering than reverse engineering, because I still feel like I'm doing art rather than science or engineering by using evolution. So could you kind of share your perspective, forward engineering versus reverse engineering? As I said, I asked the challenging question. I asked similar question to Jay Kijley and all the big names before.
Yeah, it's a great question. This project, yeah, as you say, is very fundamental. We are just trying to understand what natural mutations we can observe that will optimize the metabolism, but we're not really leveraging those here to make a product or valorize lignin, right? That's the task of new postdocs in the group. So we have a great new postdoc named Katie Maines, who is kind of taking over this now. I'm actually not funded to work on this anymore, but she is now taking some of these mutations, particularly the formaldehyde tolerance genes and that regulator that I mentioned, and putting them into our strains that can take alkaline depolymerized lignin to muconic acid. So those strains are engineered pretty well to perform in bioreactors in terms of titer rate and yield making mucinate, but there's always space for improvement, right? So she's evaluating the effect of these single point mutations in those strains, but I don't know yet if she has any results. It's kind of a strain building project at the moment.
I see. Thank you. I see more questions from some attendee. So is it question or comment? I will read this one. So other options, because highly invasive in vivo hypermutation is awesome. Mutas T7 series of method, Trident and Trace are based on that. There's also a CRISPR tilling method, Evolver, a Cas9 method, and episomal DNA method, originally AuthorLab, but a lab just came up with Bacolab. Okay, it's not the question. And then second one is Cas3, this is the correction, not the Cas9. I think it's called commuter, but with some number of lowercase letters. Okay, thank you so much for the comment. I believe I have another question from Allison. The mechanistic work is...
Maybe I could just put one comment in there.
Sure,
I was
please.
like Evolver, since that's from my group and Dave Schaefer's group.
Sure.
But I think it might actually dovetail really nicely with this talk, in that one thing I really like about this ales use and the talk was how many targets of mutagenesis you obtain that were not predictable. In all those CRISPR-based methods and the OrthoRep, those all depend on knowing what your target is. You could do fine-tune mutagenesis on that target. So I think it might dovetail nicely with this. Like you isolate different loci of interest and then could hypermutate those loci. But identifying what needs to be mutated, I think that use of ales is fantastic for that. I was very interested in your talk about you're mentioning that several of the targets you would have had no idea a priori that those would be the ones that would be mutated.
Yeah, I agree that these techniques are really nice for when you know the target, and maybe that's something we should look into actually for some of these ones that we know are kind of mutation prone in these scenarios. That's a great idea. I know Victor De Lorenzo's group has adapted the sort of like an orthorep system to Pseudomonas putida.
Right.
So maybe we'll get our hands on that and give that a whirl. Thanks.
Sure. So back to Alison's question, the mechanistic work is very interesting. The NIG-M protein, I think it was NIG-M, seems to be a structure made from threading of the homologous protein. Is there incentive or reason to obtain its actual crystal structure? That's a question.
Yeah, thanks, Allison. So Lig-M actually does have two crystal structures. They were solved at the same time by A.J. Masai's group who studies Sphingobia, and then another group in Germany, I think. And so the mechanistic discussions of that enzyme are really detailed and really well established, like in terms of the order that the substrates go into the active site and where on THF the methyl group winds up. So that's really well described. You might have been talking about Van A. That one we don't have a crystal structure for. In a totally separate project with Eugene Kowalska, we were working on some of these risky type enzymes for demethylation of guaiacol. And as part of that process, we got some crystal structures of that enzyme and tried to get one of Van A. This was with John McGeehan's group at Portsmouth at the time. And Van A just is very hard to crystallize. We were able to get a little bit of diffraction from it in order to make an AlphaFold model that was had some constraints from the crystal structure, but we couldn't solve a true experimental structure there. So maybe a task for cryo-EM or somebody else.
Sure. John, you said you have another question.
I think we actually covered it in the other questions. So
Okay, so I have one more question, but I will ask after this formal seminar. I see another one, but this is not the question. Okay, fantastic. Okay, let's close for now. So thank you so much all for joining and staying today. There will be no seminar next week due to the AICHE meeting. We'll meet again on November 16th, 2 weeks from now. Thursday, the same time, the same Zoom link. We'll have Professor Johan Parson at Harvard University and Dr. Caitlin Daly at University of Nebraska Medical Center. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks, and stop recording. Just 1 second.